Hello, today I am showing you these 3 stick sisters, Sunny, Surprise, and Sophie. The interesting thing about these sisters is that they all come from one parent, Naomi. Yes, indeed, they have no father, no sperm contributed to the making of these phasmids. In fact, they come from a long line of fatherless phasmids, all descended from my first stick insect, Twiggy. You're probably wondering why those nymphs don't look the same. Well, as opposed to the popular misconception taught in GCSE biology, parthenogenesis is not the same as cloning. Well, in very simple organisms, GCSE biology might be right, maybe in aphids or something, or not even. Anyway, the stick insects are very complicated organisms, and they were produced by a type of parthenogenesis called ootomyxis. So, you may know the process of oogenesis. If you don't, look at this or watch a different video explaining oogenesis. Anyway, it's not really that important. All you need to know is that there are 2 different types of ootomyxis. Type 1 is called, well, I just called it type 1 because that was the first one I found out about. Anyway, central fusion. And so the egg pronucleus, I think it's called a germinal vesicle or something, fuses together with the primary polar body to form a zygote. And in terminal fusion, the germinal vesicle thing fuses together with the secondary polar body. So now you're probably wondering, why don't I see people anywhere produced by parthenogenesis? Well, I really think that it would be brilliant if we could reproduce this way, but unfortunately there exist these barriers in the way that are imprinting regions. That is my definition of imprinting regions. And at this point, I introduce a new and exciting type of science called epigenetics, which explains the reasons why certain genes are silenced. And I will make a later video explaining epigenetics in greater detail if this video is popular, because it is very interesting. It is one of my greatest interests, and unfortunately I couldn't fit all of my knowledge into this short video. So yeah, that will come later. So, gene silencing. You probably know, well, you might not know, so a gene needs, in order to be expressed, it needs to be transcribed by RNA polymerase. So imagine that this is a chromatin. A chromatin is, well, it's basically DNA wrapped around histones and other proteins and RNA and stuff. And imagine that this pen is RNA polymerase. So, if the chromatin is very loose, which is called euchromatin, then it can easily be transcribed because RNA polymerase can easily get to it. But if, on the other hand, the chromatin is heterochromatin, which means it's tighter, more condensed, then RNA polymerase cannot easily get into it and transcribe it. So then, if it's not easily transcribed, then the gene is silenced. I really encourage you to read further into this topic because it's very interesting. Anyway, I'll cover the basics. So the factors that make it tighter or looser are things like methylation, acetylation, and phosphorylation. Lastly, I would like to add that while humans and other mammals cannot reproduce naturally by parthenogenesis due to imprinting regions, in the lab we can overcome the barriers of imprinting regions For example, once a mouse by the name of Kaguya was born. She was not quite a true Parthenogenesis since she had two mothers as opposed to one.